Hi, this is Xinfen Shao from University of Southern California, and I have nothing to declare. So here's the structure of the blood-brain barrier, which is very important to protect our brain from harmful substances and to maintain the healthy function. So the BBB has multiple layers, including an inner layer of the endothelial cells sealed by the TET junction and covered by layers of the parasites and astrocyte end feet. So there's a very important protein called aquaporin 4, which is the water channel, which locates along the end feet of the astrocytes. And AQP4 is very important to assist the water exchange from intravascular space into the tissue space and help maintain the brain homeostasis and glymphatic function for clearance of the harmful substances. So for example, here's a diagram of the kind of the healthy conditions. So AQP4 aligns perfectly on the end feet of the astrocytes, and the flux from the perivascular space into the tissue just carries all the harmful waste into the drainage pathway. So however, there are some disease conditions will trigger the AQE4 redistribution, which slows the waste clearance through the lymphatic system. So here's an animal study showing in the AQP4 deficient mice, you will see the increased water exchange time or the decreased exchange rate during the AQP4 deficiency. And also under the neurodegenerative conditions such as Alzheimer's disease. So here is an amyloid plaque just trigger the redistribution of AQP4, those black dots. It's supposed to be along the vessel wall, however, it's just redistributes. So you can expect a slower exchange rate from the vascular into the tissue. And also, this study shows along normal aging, you can also exploit a widespread loss of the AQP4. So those studies indicate study the blood-brain barrier water exchange rate can be potentially a very early biomarker for neurodegenerative disease. So the goal of this study is to study the age-dependent changes in BBB function being measured by the water exchange rate KW across a wide range of the age groups. So in this way, we can build up a baseline for future clinical studies of the technique. So the technique we use is based on arteriosome labeling, which label the spins from the labeling plan around the large arteries. And after a short time, when we look at the imaging volume, the signal will start exchanging from the capillary space into the tissue space across the blood-brain barrier with the exchange rate called KW. So after an even longer time, typically a post-signal delay about 1.8 seconds, you can see more spins enters into the tissue space. And this is where we get a typical perfusion image. But there's still some portion of the signal remains in intravascular space or the capillary space. So we figure out we can apply a small diffusion gradient to numb this portion of the signal because the signal in the capillary has higher velocity and also due to the pseudo-random oriented the capillary structure. So in this way, we can get a lower intensity. It's kind of still a perfusion image. And based on this image, these two images, we can apply some post-processing algorithms such as TGV regularized single pass approximation modeling to estimate how fast water exchanging from the intravascular into the tissue space. And we can generate this KW map. So this technique in our previous study, we have demonstrated it has a good test reliability. We measure from about 2 months apart from the same participants. So all MR experiments in this study were conducted on 3T Siemens Prisma scanners. We keep all the protocol identical from different research institutes, and we apply a two-stage approach for simultaneous quantification of ATT, CBF, and KW using our in-house developed MATLAB-based toolbox. So our human subjects are all cognitively normal, and with age range from 8 to 92 years, which a pretty wide range of the age. And all 161 subjects are recruited from the following 5 different groups. So here's a histogram of the age distribution of the subjects. You can see there's a generally kind of equal distribute along the different age groups. However, there's slightly higher concentration from subjects with age from 70 to 80 years old. So here I'm going to show the KW maps from 7 age groups in male and in female. So you can see the relatively constant KW in young to middle age subjects. Well, it starts declining. So here's the average KW values. Especially, you can see a large decrease of KW in the last age group. So you can see a similar pattern in the female groups. Well, the decay of the KW seems to be slower in female. So here's a scatter plots of the KW from the whole brain versus age from all the subjects. We can see the trend of decrease in KW with age is quite significant with a p-value equals to 0.004 using linear regression. So this pattern is still significant for the male group. However, the decreasing is no longer significant with a p-value 0.21 in female groups, which may indicate females are more resilient to the decline of KW with aging. So however, we also noticed there's a large spread of the KW values in subjects older than 50 years. So this may indicate some like more complex changes in the BBB function in some of those like older individuals. So when we apply a second order polynomial fitting to the same data of the KW values in whole brain versus age, we can see the change of the KW is relatively stable in young and middle age groups, while it starts declining in older subjects. So but overall, the pattern is still decreasing, and the pattern of KW decreasing with age is still, quite consistent, like in all other smaller brain regions. However, we also notice some very interesting inverted U-shaped patterns in some brain regions, such as the temporal lobe, the caudate, ACC, hippocampus, perihippocampal gyrus, and the medial temporal lobe. So in conclusion, we studied age-related KW changes in 161 cognitive normal subjects, and we observe a significant trend of decreasing KW with age, which may indicate a compromised kinetic function and slower vis clearance. And we also notice a large spread of the KW values in subjects like older than 50 years, so which may indicate compromised BBB function in some of those aging individuals. In our previous study, we noticed there's a positive correlation between KW and CSF A beta 42 and episodic memory score in the cohort of subjects also with normal combination, which indicates our technique can be a very promising biomarker to study the neurodegenerative disease. And lastly, we notice inverted U-shaped correlations in some brain regions, which may indicate a more complex brain dependent changes in the BBB function. So thanks for your attention.